Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on absorption of protein. As you can see on the screen, I am showing you the title Absorption of Protein, and at the end, I have put a question mark at the end of title. So basically, we are asking the question, what is the absorption of protein? What is the meaning of that thing, absorption of protein? See, in the previous lecture, we had discussed about the digestion of protein. Digestion of protein is taking place in the lumen of stomach and also in the lumen of small intestine. So, in the lumen of stomach, gastric juice, pepsin, pepsin act on protein, and in the small intestine, pancreatic juice, in that pancreatic proteases, then the class of protease, one class is peptidase. So this peptidase are also acting on protein. So protein is digested in the stomach, then rest of the protein is digested in the small intestine. So here you see in the lumen of small intestine, protein is digested and some of the amino acids are removed. So once the amino acids are removed, the remaining part of the protein, which is having the larger polypeptides, and these larger polypeptides are known as the oligopeptides. And in the previous lecture, we had learned that this oligopeptide, they are digested by the brush border enzyme, the peptidases. This is the brush border. Okay, and when the digestion of oligopeptides start, then there is a formation of tripeptide, dipeptide, and the amino acid. So right now you can see in the lumen what is there. So in the lumen there is a tripeptide, there is a dipeptide, and there are the amino acid. Now absorption of protein, what is the meaning of this and what is the mechanism? So here this cell, this is the enterocyte cell of the small intestine. Here this blue color space, this space is the interstitial space, and here this is the blood capillary, and in the capillary there is the blood. So the absorption of protein means the transport of tripeptide in the blood capillary, transport of dipeptide into blood capillary, transport of amino acid from the enterocyte to the blood capillary. This process is known as the absorption of protein. But it is not that all the tripeptide is directly entering into the bloodstream. It is not that all the dipeptide, they are entering into the blood of the capillary. So what happened? Some of the tripeptide and some of the dipeptide, they are digested intracellularly by peptidases. So there are different mechanisms for the transport of amino acid than the dipeptide and tripeptide. So one by one I am explaining all three mechanisms. Okay. Now first of all we are transporting this amino acid, we are sending this amino acid into the blood capillary. We are sending this amino acid, we are transporting this amino acid into blood capillary. Now what is the mechanism, how this process is taking place? So here, this is the same cell, and this one is the basolateral membrane. And in this membrane there is a sodium potassium pump is present. So here this one is the sodium potassium pump. Now what this pump is doing? So this pump is pumping sodium out. So sodium ion Na plus, they are leaving the cytoplasm of the enterocyte and they are entering into the interstitial space. So as the Na plus they are leaving the cytoplasm and they are entering into the interstitial space, what happens in the cytoplasm? The concentration of Na plus decreases. Okay, Na plus concentration decreases. So now concentration gradient is developed, that is the electrochemical gradient is developing. And how this development is taking place? So in the figure I am explaining this. So here, due to the pump, Na plus leaves the cell, and this is the lumen, and large amount of Na plus are present in the lumen. So suddenly this Na plus, they are rushing into the cytoplasm. So many Na plus, they are rushing very quickly into the cytoplasm. Now along with this sodium, okay, now the movement of Na plus, so Na plus, here the larger concentration of Na plus is present, and here the lower concentration of Na plus is present. So we can say the movement of Na plus is taking place in the direction, that is the downhill direction, from higher concentration to lower concentration. That is the downhill movement for Na plus. Okay, now together this Na plus, okay, and the flow of Na plus is so much high that this co-transporter, this is the purple color co-transporter or the carrier protein is shown. So this carrier protein, this carrier protein, the name of this carrier protein is sodium amino acid co-transporter. So this one is the sodium amino acid co-transporter, and this transporter along with the sodium is transporting the amino acids into the cell. So now as you can see here in the cytoplasm, the amino acids are shown, and here again this is the, now here the amino acid in the cytoplasm, and this one is the basolateral membrane. So in the basolateral membrane again there is a carrier protein, and what this carrier protein is doing, so this carrier protein amino acid combined with amino acid joins with the active site of this carrier protein, and this carrier protein transport amino acid into the blood capillary, that means in the blood. So this amino acid enters into the blood. So this is the absorption of amino acid. This is how the amino acids are absorbed. Now here, now let's discuss about how this tripeptide, this one is the tripeptide, and this one is the dipeptide. Now how this dipeptide and tripeptide, they are entering into the cytoplasm of enterocyte. So here you see in the figure, I am showing you in this figure, this is the transporter, and this transporter or this carrier protein is bringing peptide, that means dipeptide or tripeptide. So this carrier protein is bringing this dipeptide or tripeptide into the cell. But the question is how? What is the mechanism? So you see, due to the downhill gradient, many Na plus, they rush into the cytoplasm. Okay, that we learn in the transport of amino acid. Many Na plus are coming inside the cytoplasm, and this Na plus, they are positively charged. So to balance this charge, this is this carrier protein, the name of this carrier protein is the sodium hydrogen exchanger. So this one is the sodium hydrogen exchanger, this carrier protein is. So what this carrier protein is doing? So this carrier protein is sending the Na plus into the cytoplasm, and from the cytoplasm this H plus are transported out. So since the H plus are transported out, what happens? The concentration of H plus decreases in the cytoplasm. So here, as the concentration has decreased, what happens? This pump. Okay. Now the name of this pump is hydrogen oligopeptide co-transporter. The name of this pump is hydrogen oligopeptide co-transporter. Now what this pump or this carrier protein is doing? So here you see, now the H plus from outside, they are rushing into the cytoplasm. Okay, many hydro H plus in large number, they are entering into the cytoplasm, and along with this H plus, dipeptide or the tripeptide, they are also transported along with the H plus ion into the cytoplasm. So this carrier protein or this transporter for the peptides and the H plus, this carrier protein is known as, in short, it is known as PEP1. Okay, and the full form of PEP1 is hydrogen oligopeptide co-transporter. Okay, that is the name of this PEP1. So this is how dipeptide or the tripeptide enter into the cytoplasm. Now after the entry of this dipeptide and tripeptide, what is happening? See, now here in the cytoplasm, you see the tripeptide are transported and the dipeptide are transported. So tripeptide and dipeptide, they are in the cytoplasm, and here this one is the blood capillary, and in the capillary there is a blood. Now what happens if you observe closely this figure? Not all, few tripeptide, they leave this cell and they are directly entering into the blood capillary. This dipeptide also, they leave the cell and they are entering directly into the blood capillary. Now it is not that all the tripeptide or all the dipeptide they enter into the blood capillary. Now what happens to the remaining tripeptide and dipeptide? So the dipeptide or tripeptide which are present in the cytoplasm, on this peptidase enzymes are acting. So intracellular digestion is taking place by tripeptidase and dipeptidase. So tripeptidase act on AA3, dipeptide act on AA2. So now in the structure of tripeptide there are 3 amino acid, and between the amino acid peptide bond are there, and these peptide bonds are broken down by this tripeptidase. So due to the action of tripeptidase from the AA3, 3 amino acid are released, and due to the action of dipeptidase on dipeptide, by breaking one peptide bond, 2 amino acid molecule are released. Now you see this is the green in color. These are the carrier protein, and from the cytoplasm all this amino acid, as you can see, they are transported with the help of carrier protein. So since the transportation of amino acid is taking place with the help of carrier protein, so this type of transportation, this type of movement of amino acid from the cytoplasm to the blood capillary, this type of movement of amino acid is known as facilitated diffusion. So this one is the facilitated diffusion. Okay, now for this I am showing you one figure. See, in this figure, okay, see, this one is the hydrogen oligopeptide co-transporter, that is PEPT1, and this one is transporting dipeptide and tripeptide in the cytoplasm. This dipeptide and tripeptide, they are leaving the cell and they can directly enter into the capillary. Then the dipeptidase or tripeptidase, they can further digested by peptidases, dipeptidases or tripeptidase. So from the dipeptidase or tripeptidase, amino acid are formed. This is the carrier protein, and here this mechanism, this is the facilitated diffusion for the transportation of amino acid from the cytoplasm to the blood capillary. Here the sodium potassium pump is shown, and in this is the transportation of sodium, an amino acid. So this transporter or the carrier protein is known as sodium amino acid co-transporter. Now one more thing, this point, endocytosis. Okay, so absorption of intact protein, absorption of intact protein. Intact protein, that means the protein is not digested and it is absorbed in the same form in which form the protein was taken, and that form, or it is digested intracellularly. It is digested intracellularly. Now what they are talking about, that here this is the theory point, and here they are talking about the colostrum. Now what is colostrum? Now after the birth of baby, the mother's breast secretes the colostrum. Okay, and this colostrum is having high content of protein. Now the newborn baby, the infant, sucks mother's nipple. Okay, and it is having mother's milk. Now the baby is having the baby is getting the nutrition from the colostrum, that is the mother's milk. Now in the newborn infant, the digestive system is not very well developed, and to get the nutrition, to get the nourishment, the baby is sucking the mother's nipple, and initially, at the initial stage, in the initial few days after the birth, the baby is basically sucking the colostrum. So now what happens to the colostrum protein? So here, this is the lumen of newborn, small intestine lumen. These are the protein of the colostrum. Colostrum protein are shown, and this is the newborn baby's enterocyte cell and this cell. So this one is the enterocyte, the name is written, and this one is the M cell. Okay, so now the protein from the colostrum, 10% protein, 10% protein enters as such into the blood capillary, and 90% protein of the colostrum is intracellularly digested. So in the cytoplasm there is a lysosome, and this is the lysosome, and in the lysosome there is proteolytic enzyme, and these enzymes of lysosome, they digest the protein, and so we can say the digested protein intracellularly, it is transported. So this is the processed protein which enters into the blood capillary. Now here, okay, here in the M cell also, the colostrum protein directly it can enter into capillary, or the proteolytic enzymes of lysosome, they digest the protein. So this is the degradative pathway, and the digested protein, then they are known as the processed protein. So processed protein then enters into the blood capillary. So now, since this protein, many of the protein molecules, they are enter into the newborn's bloodstream as such. So this proteins, what the role this protein are playing? So They are providing, these proteins are providing passive immunity to the newborn. Okay, and the second thing, they are also responsible for the allergic reaction in the newborn sometimes, because the protein, the colostrum protein, which is entering into newborn's body, is acting like the antigen. That's why. So all this, and the last point, now here this point is written over here. See, whatever the food we eat, whatever the protein that we ingest, only 2 to 5% of protein we cannot digest, we cannot absorb. So we can say 2 to 5% of protein escape the digestion and absorption in the small intestine. Then this protein enters into the colon, and in the colon the bacteria are present. So the colonal bacteria, they are digesting the dietary protein 2 to 5%. So that means 100% protein is digested. So even if the 100% protein is digested, in our stool there is protein is present. So from where this protein is coming? In the stool. So in the stool, the bacteria, the cells of bacteria are present, and the cellular debris. Cellular debris means the cells of mucosa, the GI tract. In the lumen, the cells, the mucosa is surrounding the lumen. So time to time, this mucosal cell, the epithelial cell, they are shed off. So this shed off epithelial cell and the bacteria, okay, they are present in the stool. So the protein which is present in the stool is due to the bacteria and the cellular debris. Now, before I wind up this lecture, I would like to show you all the figures and all the theory points. So you go through this theory points, you read these notes, and this is the important figure for the absorption of protein. This is also very useful information. This one, this one. Second point, and the endocytosis of protein. Then the sixth point, seventh, and the 8th point, then this 9th and this figure. So with this we have completed the discussion of absorption of protein. I hope this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.